Yeah. I was being genuine. I was being sarcastic. Okay, so karyotyping, photographs of your actual chromosomes. So humans have 23, and they found that with humans, everything looked great until the end here. What they noticed with females is you had your last two chromosomes were very distinct, and they were the same for females. So your X and X pairing matched up perfectly. But then with males, if you come down here to the end, you can actually see two very distinct different types. So there's an X and a Y. And the Y chromosome is very small. And all it really does is it just determines sex. And there's like a few little things in there, but whereas, yeah. So that's all. That's the only difference between males and females is right there versus right here. Okay, is there a karyotyping question on that handed assignment that I gave out? So I can't remember if it's there or not. But so otherwise, all different pairs match up. You'll notice slight shapes and stuff like that, but that's normal. Okay, it's just based on how they extract them. Okay. So here's how karyotyping actually works. They take blood and they put it through what's known as a centrifuge, and that's basically a big dryer. Basically, it just spins and spins. No, there's no heat, but it just spins around and around and around and around. And by doing that, it actually separates everything in the blood. And you can separate the red blood cells and the white blood cells and everything from that. And when you separate the white blood cells, they'll add like a hypotonic solution to it and extract the white blood cells. And by doing that, your white blood cells actually have a nucleus, and they can actually get your DNA from your white blood cells. Because remember, red blood cells, they're actually dead, right? They get rid of their nucleus so they can carry more oxygen. Remember that from health science? So, then what they do is they prepare it, they put it under a microscope. The slide is viewed with a microscope, and images of the metaphases of the chromosomes are sorted by size and shape on a computer. The resulting display is the karyotype. So there you got 46 chromosomes or 23 pairs. There'll be 22 pairs of autosomes, which are non-sex linked, and then 2 pairs of sex chromosomes. Okay. So, and one key thing to note that I forgot to mention down here at the bottom of the slide is like the sex of a child is always determined by the male. All eggs in females have an X chromosome, right? Because that's all the mother can give it is an X chromosome. Whereas men, when they go through meiosis, when they split up their homologous pairs, you'll have an X and a Y. Not necessarily, because embryos do develop as females initially. There is a shot of testosterone that kicks in that determines. Yeah. So, yeah. Like I think I mentioned that all guys, we do have a little tiny pair of vestigial ovaries. Because, yeah, they start off as development and then it changes. So, but yeah. So it's like, like, what's her pickle? Amber Lynn. She was the first wife of Henry VIII, was it? And he had her killed because she couldn't give him a son. When it was actually his fault, right? It was his doing. So I always thought that was kind of ironic, maybe a little like, haha, but it's been a while now, so I think we can joke about Amber Lynn. I don't know, is it safe? So just to give you a heads up there. So here's 2 karyotypes. They're not on your sheet. Could you guys tell me the sex of this individual here? Man, absolutely. There's a little Y right there. Now, can you determine a problem with this one here? On the X, right? They have a third X chromosome. I think it's Turner syndrome. I can't remember. Yeah, but I can't remember the name, but I taught a student with it once. Now, basically, what it does is it's a genetic disorder. It's a chromosome disorder, so it's kind of like Down syndrome. But you have an X, so the person is typically stunted hormonally. Oh yeah, yeah, yeah. They never quite grow up. Yeah, and developmentally delayed just a little bit. The student that I had, like they were giving her shots of estrogen to kind of see what that would do to kind of maybe promote puberty in some senses and stuff like that. So yeah, so that's what that is. Okay. All right. So good. Everyone's an expert in karyotyping. All right. Our next example of biotechnology: gel electrophoresis. Okay. Phoresis. Yes. Gel electrophoresis. So what this does, it's used to separate different lengths of DNA strands so that scientists can actually study the fragments. All right? It's used in forensics, molecular biology, microbiology, and biochemistry. As a result, it can assist in genetic diagnosis of an embryo or genetic fingerprinting. So like they can use it to do for genetic diagnosis with an embryo, or they can actually use it like in forensics. They can compare people's DNA. This was kind of the first one of the original ways that they did it. It's changed now a little bit. They do different things, but this was kind of the first one. So what they would do is they would actually take a DNA sample, whether it's blood, hair, or whatever, and they would put it in a solution with these things called restriction enzymes. And what the restriction enzymes would do is they would actually chop up the DNA into smaller segments. Okay? Then they would take the DNA on this machine. You can see it here. It's like this little thing. It's literally sitting in suspended gel. You would put this glass plate in, and it has these little wells here, these little slits in it. And what you would do is you would put fragments of the DNA, of the solution with the cut up DNA, into each one of these wells. Okay, so it kind of like sitting there. And then what you do is you actually run an electric current through the gel. Okay? And what that does, so there's a positive and negative pole, the electrical current is passed through, and the DNA will actually move from one side of the plate. It'll be pulled from the wells, kind of down the glass. Okay? So it actually moves it this way. And what happens is when you randomly cut up the DNA, the smaller pieces will move down faster, whereas the bigger pieces will kind of only move so far. And then what you end up with is a little photograph like this. So these would be all your smaller pieces of DNA versus larger pieces up here. So then what you would do is you would do that with your sample, and then you would do that with someone else's DNA. And if their DNA is the same, it should cut up the same with the solution. And so when you put them together, they should be fairly similar. And then you can say with a certain amount of probability, this is the murderer, this isn't the person, stuff like that. Okay? So that's gel electrophoresis. Okay, now the next one was the Human Genome Project, or the HGP. This one was huge. Okay? This was an international research effort to sequence and map out all of our genes. Yeah, this was huge. They did this back in 2003, so this is 20 years ago, and we're still talking about it because it's leading to all of this genetic research now. This kind of like paved the way for now that we know, like we can identify genes in the human body that, oh, this is going to lead to like a 90% chance that you'll get breast cancer, or you're probably going to get Alzheimer's. Like these are certain things that we can tell now because of this project here. Okay. Mapped out all our genes, our genome of members of our species. Okay, the data was released back in April. 2 million, approximately, they looked at 2 million sequences of DNA. Okay, and that was approximately 3 billion base pairs. So, like base pairs, I'm talking about ATGC. So they looked at 3 billion of those. Yeah, the nucleotides. Very good. Yeah, one at a time. So they laid it all out, all 3 billion of them. And they're like, okay, so which ones are which gene? And they had to do that all. Massive, massive undertaking. Okay, so some suggest that there's about 1,900, or sorry, 190,000. No, sorry, that's 19,000 protein coding genes. Sorry, math's hard. Okay. There's no comma. I need the comma to space them out. They don't ever do that anymore. I know you're supposed to do. I grew up with that, so. So some suggest there's 19,000 protein coding genes in the human body. So, and other cool things that they discovered is like there's a lot of dead space in our DNA. There's like genetic coding there that we don't use anymore. There's stuff that's been passed down from like they can code now, like they can determine where like DNA came from Neanderthals. When humans and Neanderthals kind of like crossbred and everything like that, they can determine like all the viruses that have been injected into our body. Neanderthal, okay, so we're Homo sapiens. Homo Neanderthal, they're slightly smaller. They were stockier. They had a bit of a smaller brain, bigger noses. They were really adapted to living in cold weather. So you'll find them in like northern Europe, northern Germany. They were discovered in a part of Germany like Neanderthal, Germany, and so they were just a little bit slower, a little more dim. But we kind of Homo sapiens when they left Africa, they came up into northern Europe. They came across them. And what they can tell is that we lived together and coexisted for years. We bred and all that together, lived together, and then eventually we killed them. We got rid of them. Well, probably, or like competing resources. Kind of like, when you watch like monkeys in the jungle fighting over stuff, like killing each other. Probably something like that, territory and stuff like that. We used our big brains. But during that time of living together, they gave us some DNA. And a lot of it actually indicates it's like the DNA that leads to like cancer. Yeah. So that's what happened there. And this project led to all of that. Because not only now have we sequenced our DNA, we've sequenced Neanderthal DNA. They've been able to extract some out of fossil records. They've sequenced animals, all of these sorts of things. They're working on a mammoth, yeah. I believe they're going to try to clone one in an elephant here. Yeah. Yeah, they found them like frozen. Yeah. Yeah. So they'll try to extract the DNA and see if they can clone it and then have it birthed by an elephant. Yeah. I hope that doesn't happen. Oh. I don't even know. I don't know. I don't know if there's any DNA kicking around of a preserved dodo bird. That's a good question, though. I've never thought about bringing back the noble dodo. Oh, it won't be like Jurassic Park or anything. Yeah, they're just a bigger elephant. They're not like 8 stories tall or anything like that. Yeah, but a lot of people will pay a lot of money to go see a woolly mammoth. So that's gonna win out. All right. Um, no, no, no. Like, They lived on like woolly mammoths and mastodons. Like that's another type of furry animal. They lived on like the plains areas. Well, not here, but like during the ice age, like down in the southern states where it would have been kind of like this now. Um, yeah, yeah. Like it would have been dramatically cooler for them. Big things that changed for them though that wiped them out was like, well, one, humans showed up in North America and we got lots of them. But also, there was another shift that wiped out a lot of animals that lived around here, like the rhinos and everything that we had. Because we switched from like we just used to be all forest and trees and everything, so they ate leaves. And then with the climate that changed, just turned into a grassland and they couldn't eat grass and helped wipe them out too. So there's a bunch of things. Yeah, we got rid of the. I was just gonna say the giant sloth that was like the size of a grizzly bear. Yeah, they were massive. Yeah, they eat plants, trees, but they were again big, slow. So they were big and slow. So they were easy to hunt, right? Same with woolly mammoths and mastodons. So bigger, yeah. They'd be like 9 feet tall. Yeah. Yeah. So so this is just a picture of what some of the mapping looks like, and like some of the names for some of these. Okay, so like factor Y deficiency, CR1 deficiency, factor H deficiency. Like there's a ton and ton and ton and ton of these. So yeah. Oh yeah, we're recording this. We should keep going for sound. Okay, so mutations. All right, so let's get into what happens when genes mutate. So they're caused by mutagens or mistakes during DNA replication that aren't fixed. Okay, so it's not like on a Marvel comic or something like that where an A bomb goes off and all of a sudden you get superpowers. It's basically you're exposed to something and it causes your DNA to replicate and make mistakes. Okay, so for example, the wrinkled phenotype of a pea seed is caused by a a mutation in the gene that codes for an enzyme that influences the shape of the starch molecules in the seed. So wrinkly peas are actually caused by a mutation. All right, so all that really happens is I'll show you guys here in a minute. Is like basic when you talk about like remember like when you're replicating DNA, the DNA uncoils and you lay out your base pairing, you accidentally put the wrong parent, the pairing in, and it just codes a little bit differently. All right. Mutations can range from changes in a single base pair in the coding sequence of DNA to actually a large deletion of pieces of chromosomes. And that's when you start to notice them. Any permanent change to an organism's DNA is known as a mutation. So mutations are significant because they can lead to genetic disorders. They actually also can lead to evolution, okay, which we'll get into later. So they can take place in two places. In your somatic cells, when that happens, a mutation in your genes, in your somatic cells, so your non-sex cells, it affects you. Best examples of this would be cancer. Okay, you introduce a mutagen or carcinogenic to your body, say like cigarette smoking for 40 years, you're going to cause a mutation in your genes, you're going to get cancer. Okay, that only affects you, right? If it happens in your gametes, so in your sex cells, this mutation can then be passed on to your offspring. Okay, and this is where it gets a little more complicated. Oh yeah. absolutely. There are known genes that they can look at now and figure that out for some cancers. Yeah, yeah. Like that's one of the worst things about cancer is there's just so many wide causes and there's so many varieties out there that it's so hard to know. But they can do some testing now. Mutations can be classified now as either one or two genetic types. There's either gene mutations that happen to the gene itself or a chromosome mutation. And this is where an entire chromosome is changed. Okay, much more dramatic because a chromosome, remember, contains hundreds, if not thousands, of genes. So you can alter quite a bit on one chromosome. Oh, anyone want to take a guess what the blank is that are the ultimate source of genetic variation in a population? What did we just talk about? Not genes, not chromosomes, but mutations. Thanks, guys. Yeah, mutations. Like when I was in high school and we talked about genetic mutations, it was downplayed as something that doesn't really happen. Like when it happens, don't worry about it. It's not a big thing. But now, now that we've mapped out the human genome, we know, wow, most of the things that have happened through human evolution have been through mutation. All right, and it's been a lucky, like either a good mutation or a bad mutation. Well, ideally, it has to be good for it to be passed on, and then just the environmental factors or living conditions at the time. Okay, so there are 1, 2, 3, 4, 5 types of gene mutation. Okay, to go through here. So what this does is this is going to explain what happens here. Okay, so this is a normal person. Now instead of using base pairs, what I did is I just took a sentence. The big fat cat ate the wet rat. Okay? What I'm going to do is I'm just going to change it ever so slightly every once in a while to kind of highlight the differences between these mutations. So, the first one is a missense substitution. So this is one base is changed in a codon, so it codes for the wrong amino acid now. So just one thing, remember, one wrong part. Because remember when I talked about protein synthesis and the unwrapping and everything like that? We're only going to switch one, like A or C or G. And that's going to just make one amino acid different in the protein. All right? Yeah, just one. Yeah. So here, the biz fat cat ate the wet rat. So all I've done is just changed one little thing. So all this does is kind of cause a change in the protein itself. It could be minor, it could be major. But associated diseases with this one would be like sickle cell anemia. So you're still making red blood cells, but they're instead of round, they have that sickle shape. Color blindness, you can still see, you just can't distinguish between red and green. Or hemophilia, your blood doesn't quite clot as well, or at all. Okay? Now, so a nonsense substitution. This is where you change one base pair again, but what you actually change to is the stop codon. Because remember, there is that one part on the wheel that actually just says stop, and it stops. That's why the sequence knows to stop, and you stop making the amino acid. Yeah, right? So it stops at rat. So here, all that happened is that I changed the R, the F in fat to the R in rat. So I'm coded for stop. So what happens is I just deleted, I just took out, 1, 2, 3, 4, 5 words. Okay. So it stops, and all of a sudden everything kind of just ends. So it's a much bigger mutation. You're going to notice this one a little bit more. An example of this would be like muscular dystrophy. All right, this is where your muscles break down in your body your muscles kind of start to shut down. Yeah, yeah, yeah. And I can't remember if it's only for your skeletal muscles or for smooth muscles as well. I think it's the skeletal. You'd be in, yeah. It's a very short timeline. I can't remember now. So the next one is deletion. Okay. So this is the deletion of a nucleotide to the changing, to change the reading frame. So here, what happened is, like, so the big fat cat, right, was my original word. Now what I ended up doing here is I took the E out, right? Instead of the big fat cat, it just shifted everything, didn't it? You guys see how that happened? One E got taken out and it caused everything else to kind of shift over to make up for it. Okay. Now this changes like every protein now that's being sequenced. And this is where you can get cystic fibrosis. Okay. Oh yeah. Yeah. Oh yeah. Yeah, and there's ranging amounts of severity. Like your cousins doesn't sound actually too bad. Yeah. 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 Do you guys remember that Robert Latimer case we talked about in Health Science, like at the beginning in September? There's the guy that killed his daughter. His daughter has cystic fibrosis. No, cerebral palsy. Sorry, never mind. Ooh, wrong one, wrong one. But it can be, yeah, like it can, like if you have too much mucus, it can, yeah. Very poor quality of life. Or yeah, drowned, need a lung transplant. Oh, I can't believe I went there. Cystic fibrosis. Sorry, guys. Ooh. I was like, oh, that's Robert Latimer's. No, never mind. Yeah, cystic fibrosis. So yeah, it can be very serious as well. Now, the next one is insertion. Okay, so it's kind of the opposite. So here, the addition of a nucleotide that causes the shift. So here you put another piece in, and it pushes everything else down. So here, this Z got inserted. I almost said inserted. All right, and it actually then causes everything down the line to change. Oh yeah. Yeah, like, well, it'd be for the genes that are expressed in the intestinal tract, yeah. So just those ones. It could be throughout his entire body that shift happened, but it would be there that you would notice it. Yeah. So, yeah. So Crohn's disease, colitis is another one like that. These are all inflammation of the intestinal tract, and sometimes people are just prone to it. Like it's just you're genetically you get this hand of genetics that eventually this shift will happen, or this insertion will happen, or more likely to happen. Okay. All right. So the next one is expanding mutation. And this is tandem repeats of codons expand from generation to generation. So as it gets passed down, the codon just keeps getting bigger and bigger and bigger and bigger. So generation 1 would have the big fat cat ate the wet rat. Generation 2, so their children would have the big fat cat ate the wet rat. And then number 3, the third generation, the big fat cat ate the wet rat. So it just keeps growing and growing and growing. Yeah, like this is a bad one. Because as it goes down the line, it gets worse and worse. So typically now, people that it's been in their family for generations, it's quite severe. Do you guys know what Huntington's disease is? It's basically a death sentence. It's kind of like Lou Gehrig's disease. your body just kind of starts to shut down, and you can slowly become more and more incapacitated, and things start going offline, and then eventually, um, yeah, yeah, yeah. It's 100% like they can test you for it now. Like if like, let's say your mom had it, there's a 50-50 chance you're gonna get it. Yeah, yeah. So so they test you for it now if you have it in your family. And like I've listened to or like radio shows and stuff like that where they'll interview children of parents that have Huntington's, and then like they get tested for it, and they know one day they're gonna get it. So it's like, well, do I have children now, or what do I do, and all these sorts. So it's pretty nasty. It really does weigh on people a lot in psychological sense too. Yeah. Oh, I see. Like, is it like doubles or triples or something like that? Yeah, yeah. I'm not sure if it's a sequenced amount every time or not. That's a good question. But yeah, I'm not sure. I'll look into that. Oh my gosh. You gotta go? Okay. I can send you this video too. If yeah, no problem. Okay. So what's this? So genetic disorders caused by gene mutation. There's actually three main types of genetic disorders. So there's recessive genetic disorders, dominant, and then there's sex-linked disorders too. And again, we talked about all of these before. They just pass down like anything. Okay. So the first one here is cystic fibrosis. It's recessive. The gene that codes for a membrane protein is defective due to a deletion mutation. Affects excessive mucus is produced, and it can lead to just digestive and respiratory failure. Okay, do you guys have this? Yeah, is it okay? Oh, good. Oh, good. That one's typed in for you. No, we're kind of running behind time, so let's just cut to the answers. I'll give you guys a minute now to type these all in. So then there. Well, just get the highlights. Come on, I barely make you take notes all year. All right, so here's the last ones. All right, now the last couple slides. Chromosomes mutations. Okay. So this is not just when a gene has been affected, but the entire chromosome itself. And it's way more drastic effect on an organism because compared to a gene mutation, because many genes are involved in each chromosome. So there's four types of chromosome mutations. There's translocation, and this is where a chromosome detaches from one chromosome, sorry, a piece of a chromosome detaches and moves into a new position on that chromosome, or to possibly a whole new chromosome altogether. So you can actually rip off a piece and move it around. Now, deletion is when that chromosome breaks off and it's lost completely. It's just gone. So you could lose a chromosome. Duplication, like the one I showed you guys. So this is where an extra copy of genes are generated on a chromosome. So this would be an example, like you have the same gene or chromosome even, like that XXX one that I showed you guys. That's a duplication. Now, non-disjunction chromosome mutations. This is probably the most common. This is where some or all chromosome pairs fail to separate during meiosis. Okay, so during meiosis, your homologous pairs are supposed to separate, right? And the one from your mom, one from your dad go separate ways. Sometimes in meiosis, both go one way. So you'll all of a sudden end up with two chromosomes for one pairing to pair up with one, or you'll end up, if both go this way, nothing over here. Okay? And that's another way you can end up with 3X chromosomes or something like that. Okay? So this is where some or all chromosomes fail to separate. Gametes and new individuals could end up with higher or lower chromosome numbers than is normal. So that's typically what you'll see. All right? So in plants, this happens quite frequently. They call it polyploidoid. Polyploidoid. Yeah, there we go. These plants often survive and can actually be more vigorous in growth. Some situations in humans is usual, but usually not a good thing. In fact, most animals, we call this polyploid. Ploid. Oh, whatever. This non-jurisdiction mutation basically fail to survive, so they have this extra chromosome, or have shortcomings in structures or in behaviors. Okay? So, here's what I was talking about. Normal meiosis. This would be two non-jurisdiction meiosis examples. So it can happen in the first round of meiosis or even in the second division. Okay? Now you guys have one more table that looks like this. So the big ones are Down syndrome, Turner syndrome, and Klinefelter syndrome. Okay, that you kind of probably heard about. And here's the answers here. Klinefelter syndrome. This is the 3X. Oh no, sorry, that's the wrong one. So Down syndrome, probably the most common one that we've all heard about, right? So these are all non-disjunction ones. So this is basically an extra pairing on the 21st chromosome. So Turner syndrome, sorry, I got these backwards. This is where the female is actually missing an extra chromosome. Yeah. I'll have to look up the 3 X chromosomes. That's one too. And then Klinefelter syndrome, this is males with an extra X chromosome. So you can just copy these down.